Just a reminder to take the quiz when you are finished watching the lecture and taking your notes. And this is found on Edline in the assignments folder as well as posted on your calendar. We know that DNA is passed on from parents to children and holds all our genetic information. But how does that string of A's, T's, C's, and G's make us who we are? It's because short segments of DNA called genes code for proteins, and it's those proteins that give our hair and skin its color, make our muscles work, control the chemical reactions that break down some molecules and build up others, and let things in and out of our cells. The process of making these proteins is called protein synthesis, and it's done in 2 steps, transcription and translation. Back in Chapter 2, you learned that proteins are polymers made of monomers called amino acids, which you'll see down here in the picture. So this big long snake is the chain of amino acids called a polypeptide, and each individual red bubble is an amino acid. And this protein is constructed in 2 steps. First of all, DNA is such an important molecule it cannot leave the warm, fuzzy, protective environment of the nucleus. It has to stay there so it doesn't get damaged. So the message coded for in the DNA gets written into another language, the language of RNA. The RNA can freely move in and out of the nucleus. Proteins, on the other hand, are made in the cytoplasm on organelles called ribosomes. Once the RNA brings the message to the ribosomes, that message needs to be translated into a sequence of amino acids which will then fold together, forming a protein. So in order to get from the DNA sequence to a protein, a group of molecules called RNA act as sort of middlemen. RNA is another type of nucleic acid like DNA, so it can transmit genetic information as well. DNA transmits genetic messages from one generation to the next, and RNA transmits genetic messages within the cell. Because DNA and RNA have similar functions, they also have similar structures. RNA is also made up of nucleotides composed of a 5-carbon sugar, nitrogen base, and phosphate group. However, instead of 2 strings of them being attached in the center, RNA is single-stranded. It also has a slightly different sugar and 1 different nitrogen base. RNA contains the base uracil, while DNA contains the base thymine. In doing its job, RNA binds to 1 strand of DNA, so A's on DNA will bind to U's on RNA. The other bases will bind the same way as they always do. T's on DNA bind to A's, C's bind to G's, and G's bind to C's. So here's how the message coded for in DNA ultimately gets made into a protein. The process has 2 steps, transcription and translation. Transcription makes the mRNA blueprint, and translation takes that blueprint and uses it to build a protein at the ribosome. Transcription itself has 2 basic steps. First, DNA is unwound. RNA polymerase is the enzyme that unwinds the DNA and also adds on RNA nucleotides. Let's take a closer look at the mRNA molecule that was just created in transcription. mRNA is sometimes called the genetic code. This is because it acts like a coded message. DNA creates the code and the ribosomes then have to decode it. Coded mRNA gets read in groups of 3 RNA bases called codons, and each codon stands for 1 amino acid. Since there are 4 different RNA bases, A, U, C, and G, and codons are 3 base sequences, there are 64 different combinations of bases that can make up 1 codon. However, there's only 20 amino acids, which means that each amino acid has a few different codons that code for it. So we could say that the genetic code is redundant. If we had a list of codons, we would see the amino acids repeated as we went down the list. And this is really important because if there's a small mutation, it won't necessarily change the amino acid, so it won't change the protein. The genetic code is also universal. The same table of codons and amino acids can be used for any organism. Humans, plants, bacteria, you name it. So once the mRNA has been created in the nucleus, it goes out to the ribosome where the message is translated into a sequence of amino acids. This process is called translation. Another type of RNA called tRNA plays an important role in translation. You can see that tRNA actually looks like a lowercase t. The top part of the T contains a 3-base sequence that's complementary to the codons, and on tRNA this is called the anticodon. The other part of tRNA has an amino acid attached to it. During translation, mRNA attaches to the ribosome, and as it moves across the ribosome, tRNA is recruited to bind with the mRNA codons and attach their amino acid to the polypeptide. This picture shows a good summary of the entire process of protein synthesis. A small piece of DNA unwinds in the nucleus, and RNA polymerase adds on RNA bases to make mRNA. This mRNA then leaves the nucleus and attaches to the ribosome. The ribosome recruits the appropriate tRNAs, which bind the anticodons on the tRNA to the codon on the mRNA. As the tRNA binds to the mRNA, it attaches the amino acid on the other side to form a to add an amino acid, forming a polypeptide chain. Now you try using this picture on the slide and practice explaining how proteins are made.